Pepsin digestion. The antibody was digested with another proteolytic enzyme, pepsin, and it was observed that only one fragment of high molecular weight of about 100 kDa was produced, which was comprised of two Fab-like fragments that show antigen binding activity and were therefore named F-Ab2. The Fc fragment was not obtained due to the pepsin digestion into the multiple fragments. Mercaptoethanol treatment. Chemical treatment was done using mercaptoethanol, which helps in reducing the disulfide bonds irreversibly. It was found that 4 chains were produced. 2 of them were identical and had a molecular weight of about 50 kDa each, whereas the rest 2 were also identical and had a molecular weight of about 25 kDa each. The former chains with the high molecular weight were termed the heavy chains, whereas the other 2 were named light chains. 5 types of heavy chains. The chain structures of these antibody classes are shown below, and the properties of these are mentioned. To aid the protection against the diverse microbes, various subclasses of different types of antibodies have evolved. For instance, Ig has 4 subclasses: Ig1, Ig2, Ig3, Ig4. There is around 60% homology between the classes of antibody and around 90% homology between the subclasses of each type of antibody. Ig. It is the most abundant immunoglobulin in serum, 80% of total serum Ig, and has a molecular weight of about 150 kDa consisting of 2 gamma heavy chains and 2 kappa lambda light chains. Due to the slight differences in the gamma chain, which affects the biological activity of the molecule and decreases average serum concentrations, the subclasses are numbered Ig1, Ig2, Ig3, Ig4. All these subclasses of Ig except Ig2 can pass through the placenta, which Figure 4.110 structure of immunoglobulin is revealed by chemical and enzymatic methods. Domains of the immunoglobulins. The intrachain disulfide bonds are formed in both heavy and light chains, which creates a loop of around 60 amino residues. These loops are present in the functional units called domains of around 110 amino residues, which are present in both the chains as independently folded regions of the repetitive sequences. Therefore, there is one variable domain each in both the chains, which is referred to as VL and VH. Similarly, there is one constant domain in light chain CL, whereas there are 3 constant domains in the heavy chains CH1, CH2, CH3 in most antibodies except IgM and Ig antibodies which have 4 constant domains in their heavy chains CH1, CH2, CH3, CH4. Antibody classes. Different classes of antibodies have evolved due to the diversity in the biological properties and the entry routes of different microbes. 5 types of antibodies are isotypes: IgM, Ig, IgD, and Ig are found in mammals due to the provides immunity to the developing fetus. These isotypes are also able to bind to certain receptors on the phagocytic cells, which help in the opsonization. Except Ig4, these subclasses also help in the activation of the complement system. IgA. It constitutes 10-15% of total Ig in serum, having 2 alpha heavy chains and 2 kappa lambda light chains with a molecular weight of 170 kDa. They form the major part of external secretions like saliva, colostrum, tears, mucus of gastrointestinal, genitourinary and respiratory tracts. It is present in serum in monomeric form, whereas in external secretions in dimeric or tetrameric form. The secretory IgA that is, the IgA of external secretions also consists of 2 more poly. These isotypes are also able to different classes of antibodies have evolved due to the diversity in the biological properties and the entry routes of different microbes. 5 types of antibodies are isotypes: IgM, Ig, IgD, and Ig are found in mammals due to the provides immunity to the developing fetus. These isotypes are also able to bind to certain receptors on the phagocytic cells, which help in the opsonization. Except Ig4, these subclasses also help in the activation of the complement system. IgA. It constitutes 10-15% of total Ig in serum, having 2 alpha heavy chains and 2 kappa lambda light chains with a molecular weight of 170 kDa. They form the major part of external secretions like saliva, colostrum, tears, mucus of gastrointestinal, genitourinary and respiratory tracts. It is present in serum in monomeric form, whereas in external secretions in dimeric or tetrameric form. The secretory IgA that is, the IgA of external secretions also consists of 2 more polypeptide chains, secretory component and J-chain polypeptide. The J-chain peptide helps in the polymerization of serum IgA and secretory IgA through disulfide bridges. Secretory component is synthesized by the epithelial cells of the mucous membranes and binds to the Fc region of the IgA dimers. It is a part of the polymerization immunoglobulin or poly-Ig receptor, a transmembrane glycoprotein which is expressed in the mucosal epithelia and helps in the stabilization of the secretory IgA against the proteolytic degradation. It is so when polymeric Ig binds to this poly-Ig receptor which forms the IgA receptor complex and reaches the luminal membrane by the transepithelial transport. After the enzymatic cleavage of the poly-Ig receptor from the membrane, the transmembrane and cytoplasmic domains remain attached to the epithelial cell, whereas the extracellular domain becomes the secretory component which is bound to the polymeric IgA and is released with it together into the mucous secretions. IgA antibody provides the first line of defense against pathogens at mucosal surfaces and also provides a major source of protection to the neonate against the microbe invasion. IgM. IgM constitutes 5-10% of total Ig in serum, having 2 micro heavy chains and 2 kappa lambda light chains with a molecular weight of 180 kDa in a monomeric form. It is the first antibody that is produced by the B cell and expressed on its surface. The monomeric form of this antibody is expressed on the surface of the B cells and acts as an antigen receptor. IgM constitutes 5-10% of total Ig in serum, having 2 micro heavy chains and 2 kappa lambda light chains with a molecular weight of 180 kDa in a monomeric form. It is the first antibody that is produced by the B cell and expressed on its surface. The monomeric form of this antibody is expressed on the surface of the B cells and acts as an antigen receptor. It is also secreted by the plasma cells and exists in the pentameric form, 900 kDa, which consists of 5 units. Each unit is composed of 2 heavy chains and 2 light chains, which are joined together by the joining chain or J chain and additional disulfide bonds. In this polymeric form, the Fc regions of the 5 units are arranged in the center while the 10 antigen binding sites are on the periphery of the molecule. Hence, higher valency is seen in the case of serum IgM as compared to other isotypes, making it very effective in the elimination of the pathogen. Due to their large size, they are also found in intercellular tissue fluids. They are more effective in the complement activation as compared to the Ig due to the close proximity of the Fc regions. Due to the presence of the J chain, they are able to bind to the receptors of the secretory cells, which help them to enter external secretions and therefore, as Ig is majorly present in these secretions, IgM plays an important accessory role as a secretory immunoglobulin. IgD. It constitutes 0.2% of total serum immunoglobulin, and its serum concentration is low, around 0.3 mg per milliliter. It is also expressed on the surface of B cells, acting as an antigen receptor. When exposed to the antigen, it also helps in regulating the B cell function. Ig. The concentration of Ig in serum is very low, which is 0.3 micrograms per milliliter. It plays an important role in mediating hypersensitivity reactions to protect the host from allergic reactions and parasitic infections and causes some symptoms such as asthma, anaphylactic shock and hay fever. After encountering an antigen, plasma cells secrete Ig, the Fc region of which binds to its specific receptors on the basophils and mast cells. When the antigen is reintroduced, it binds to the antigen binding sites of Ig bound to the mast cells and basophils and therefore helps in the degranulation of these cells, causing them to release pharmacologically active agents like histamine, which gives rise to allergic manifestations. IgM Ig IgD. Type of heavy chain micro gamma alpha epsilon delta. Molecular weight. Dalton's 90000 15000 15000 Percentage of total. Antibody 6%, 80%, 13%, 0.002%, 1%. Process placenta no yes no no num. Apart from the antigen binding that is recognized, the antibodies also elicit responses or effector functions that help in the elimination of the pathogen and killing it. The variable region of the antibody helps it in binding to the antigen, whereas the constant region of the heavy chain helps it in interacting with other proteins or cells that help it to participate in performing the effector functions. Opsonization. The molecules like antibodies such as Ig that enhances the process of phagocytosis are called opsonins, and the process is called opsonization. There are various Fc receptors on the surface of the phagocytic cells like macrophages or neutrophils, which binds to the Fc region of the antibody. This initiates the signal transduction pathways through which the antigen-antibody complex is phagocytosed. Therefore, it helps the phagocytic cells to capture the antigens and ingest them where they undergo destructive processes like oxidative damage and enzymatic digestion. Antibody-dependent cell-mediated cytotoxicity (ADCC). The antibody-coated target cells bind to the Fc receptors present on the surface of natural killer cells to kill the target cells through cytotoxic activities. Here, the antibody acts as a newly acquired receptor which makes the attacking cell recognize and kills the target cells. This process is called antibody-dependent cell-mediated cytotoxicity. Toxin viral neutralization. Sometimes, antibody alone helps in neutralizing the virus and the toxins which are produced by some bacteria. They do so by binding with the part of the toxin or virus, which is important for its biological activity, or by binding tightly to the toxin or virus, thereby preventing their interaction of them with the cell surface receptors. This process is called neutralization and depends largely on the specificity of an antibody. IgM and IgA are the antitoxins that are able to neutralize the toxins. Complement activation. Complement system comprises soluble proteins, which are synthesized in the liver and then circulate in the blood plasma in an active state. These proteins, when bound to the antibody, get activated, which help in the bacterial lysis. These proteins are also activated by some components of pathogenic organisms, resulting in a controlled enzymatic cascade to destroy the pathogens. IgM and Ig helps in the complement activation. Antibodies show transcytosis. The antibodies cross the epithelial layers of cells in order to reach the mucosal surfaces like IgA, IgM, or from mother to fetus like IgA and therefore show the process of transcytosis. The properties of the constant region of the antibody determine its capacity to be transported. Antigenic determinants on immunoglobulins. Antibodies can themselves generate an immune response and act as an immunogen. These are glycoproteins. There are mainly three categories of antigenic determinants or epitopes on immunoglobulin molecules. Isotypes. As we know, based on the constant region sequence of the heavy chain, there are 5 classes of antibodies: Ig, IgM, IgD, IgA, and IgA subclasses. These are isotypes as they are constant region determinants of heavy chain and differ in the biological properties, functional locations, and ability to deal with different antigens. All members of the same species have the same isotype or constant region sequence, whereas different species will express different isotype or constant region sequences. An immune response is generated when an antibody of one species is injected into the different species as they are recognized as foreign. Allotypes. All members of the same species have the same isotypic gene, but immunoglobulins also contain genetic markers on them, which are different in different individuals. Due to the genetic differences between individuals, the same gene has allelic forms. These forms are the result of small amino acid differences in the constant region of the lighter heavy chain of an immunoglobulin. Some members of the same species will have these on the Ig, whereas it can be absent in others. These, therefore, will act as an immunogen if injected into an individual whose immunoglobulin lacks the allotype. Figure 4.111 schematic representation of the isotype, allotype, and idiotype. Idiotype. The variable regions of both the heavy and light chain of an antibody have a unique amino acid sequence, which acts as an antigen binding site as well as antigenic determinants. The same clone of B cells producing the antibodies will have these unique antigen determinants on their variable region or idiotype. The antibodies which are not derived from the same clone of the B cells will have different idiotype. Individual antigenic determinant of variable region is called idiotope, whereas a total of all idiotopes present on the antibody is called idiotype of an antibody. Monoclonal antibodies and hybridoma technology. Individual antigenic determinant of variable region is called idiotope, whereas a total of all idiotopes present on the antibody is called idiotype of an antibody. Monoclonal antibodies and hybridoma technology. Array of B cell clones from a single B cell is produced when exposed to the antigen having different types of epitopes. Therefore, multiple antibodies are produced, each specific for a particular epitope. Hence, the resulting antisera consists of a mixture of antibodies which are specific against the different epitopes of the antigens and are referred to as polyclonal antibodies. These polyclonal antibodies then help in the removal of that antigen and increase immune protection in vivo. However, due to the heterogeneity of these antibodies, the efficacy of the antisera is reduced for in vitro purposes such for research. Therapeutic uses as a single clone of the antibodies are preferable. Hence, the antibodies which are produced by a single clone of B cell and specific against a single epitope are referred to as monoclonal antibodies. In 1975, George Kohler and Cesar Milstein established a method for the production of monoclonal antibodies and referred to it as hybridoma technology, in which they fuse the antibody-producing B cell with the myeloma cells producing the hybrid cells called hybridomas, which possess the property of dividing indefinitely and secreting the antibody produced by the B cells. Fusion of the antibody-producing B cells and myeloma cells. Antibody-producing B cells are isolated from the spleen after a few weeks when they start differentiating by immunizing the mouse with the antigen of interest. Since the half-life of antibody-producing B cells is short, these cells die after several weeks in vitro. Therefore, they are fused with myeloma cells which are immortal. These myeloma cells lack the HGPRT gene, hypoxanthine guanine phosphorybosyltransferase. Therefore, the resulting hybrid cell line is produced, which has the property of synthesizing the antibody as well as will grow indefinitely. These hybrid cells contain the HGPRT gene as this comes from the functional antibody-producing B cell. Selection of the hybridoma cells. Since some antibody-producing B cells or myeloma cells are left as they are not fused, the culture plate will contain 3 cell lines, that is, antibody-producing B cells, myeloma cells, and hybridoma cell lines, which are separated using the selective medium. The selective medium is hypoxanthine, aminopterin, and thymidine, in which the hybridoma cells will survive, whereas the myeloma cells will die. The unfused antibody-producing B cells will die naturally as they are mortal or have a shorter lifespan. Nucleotide and thymidylate synthesis are essential for cell survival, and there are 2 pathways for it, either de novo synthesis or the salvage pathway. Normally, the animal cells follow a de novo pathway for the synthesis of nucleotides and thymidylate. Since the de novo pathway requires tetrahydrofolate, HAT medium containing aminopterin, which acts as an antifolate, blocks the activation of tetrahydrofolate and thereby blocking the nucleotide synthesis by the de novo pathway in all the cells. Therefore, hybridoma cells, unfused myeloma cells, are not able to synthesize the nucleotides by the de novo pathway. As mentioned above, there is another pathway for nucleotide synthesis, which is the salvage pathway. But this pathway requires HGPRT enzyme. Myeloma cells are engineered to lack the HGPRT. Therefore, they will not be able to synthesize the nucleotides by the salvage pathway also and will ultimately die, whereas hybridoma cells contain HGPRT as this comes from the functional antibody-producing B cells during fusion and helps in their survival. The HAT medium also contains hypoxanthine and thymidine supplements which ensure an adequate supply of substrates for the hybridoma cells to follow the salvage pathway. Therefore, this results in the selection of the hybridoma cells. Figure 4.112 Generation of monoclonal antibodies. Polyclonal antiserum containing the blend of monoclonal antibodies is obtained in response to the complex antigen having 4 epitopes. On the contrary, monoclonal antibodies are obtained, which are developed from a single plasma cell which is specific for the particular epitope on the complex antigen. Antibody engineering immunotoxins. These are the drugs which are protein-based and have target-specific monoclonal antibodies attached to the lethal toxin. The toxins have 2 functional domains. One is the target-specific binding domain which allows them to bind to the specific target cells, and the other is the cytotoxic domain which helps in killing the cells after internalization by inhibiting the protein synthesis. In immunotoxin, the target-specific binding domain of the toxin is replaced by the monoclonal antibody, which is specific for a particular tumor cell. It will bind to that particular tumor cell with the help of a tumor-specific monoclonal antibody and will help in killing it with the help of the cytotoxic domain, which will enter the tumor cell and will inhibit the protein synthesis, thereby killing the tumor cells. The toxins which are used in the preparation of immunotoxins are ricin, diphtheria toxin, and shigella toxin. Chimeric and humanized antibodies. Most of the monoclonal antibodies are produced in mice and are useful for research and diagnostic purposes. However, as therapeutic agents, these murine antibodies are not considered ideal as these are recognized as no irreversible chemical alteration occurs in either antigen or antibody. The interactions between the antigen and antibody are weak, non-covalent, and consist of van der Waals interactions, hydrophobic interactions, electrostatic interactions, and hydrogen bonding. These occur over short distances, and since these are weak interactions, the huge number of such interactions helps to make the strong binding between antigens and antibodies. With the help of these interactions, the binding occurs between the epitope or antigenic determinant of the antigen and the complementarity determining regions or paratop present on the variable regions of an antibody. The number of binding sites per antibody molecule is referred to as valency, and the number of epitopes on the surface of an antigen is termed valence. The antigen having one epitope is referred to as monovalent, whereas the one with multiple copies of the same epitope is termed polyvalent. Strength of antigen-antibody interactions. Affinity of an antibody refers to the total strength of the non-covalent interactions that exist between a single epitope and a single antigen binding site paratop. It is a quantitative measure of the binding strength. Therefore, low-affinity antibodies mean that there are weak interactions between antigen and antibody and are able to dissociate readily, whereas high-affinity antibodies mean that there are stronger interactions between the two and remain bound longer. Avidity refers to the overall interactions that exist between the multiple, repeating epitopes or multivalent antigens and antibodies with multiple binding sites. Therefore, it is the total of the affinities of all the epitopes involved. It is a better measure of its binding strength within the biological systems than the affinity. Pentameric IgM, which has a low affinity and has 10 antigen binding sites, has higher avidity compared to the antibody which has higher affinity and lower avidity, which helps IgM to bind effectively to the antigen and compensate for the lower affinity. Non-self by an individual's immune system will induce an antibody response which will help in clearing these antibodies from the bloodstream. Therefore, to avoid this, human monoclonal antibodies are used. But developing the human monoclonal antibodies is difficult. Hence, recombinant DNA technology is used to engineer the monoclonal antibody and antibody binding sites, which has led to the generation of the chimeric and humanized antibody. In chimeric antibody, the recombinant DNA is cloned, which contains the variable region sequence from the mouse antibody gene and the constant region sequences from the human antibody gene. This recombinant gene encodes the antibody, which is a mouse human chimeric antibody where the variable region of mouse DNA determines its antigen specificity and the constant region of human DNA determines its isotype. These are very less immunogenic as compared to the mouse monoclonal antibody. In humanized antibodies, only the complementarity determining regions are of mouse origin, and the rest all is a human antibody. Therefore, it also helps in complement activation of Fc receptor binding as the biological effector's function of the antibody is retained. Heteroconjugates or bispecific antibodies, as the name suggests, two different monoclonal antibodies are fused by either chemical crosslinking or by producing them in hybridoma. As a result, the bispecific and monospecific antibodies mixtures will be produced from which the bispecific antibody will be purified. To kill the tumor cells, in some antibodies, half of the antibody shows tumor specificity whereas the other half shows specificity for surface molecules present on an immune effector cell such as NK cells, macrophages, cytotoxic T lymphocyte which will activate these effector cells when bound to the tumor cell thereby killing it. Antigen-antibody interactions. Antigen-antibody interactions are similar to enzyme-substrate interactions except that Figure 4.113 viral neutralization showing avidity and affinity valence. Cross-reactivity. Some antigens possess a common epitope. These epitopes can be identical or similar, which are present on different antigens. Therefore, even though the antigen-antibody interactions are very specific, sometimes an antibody which is invoked by a particular antigen cross-reacts with the different antigens as they share a common epitope. The strength of the antigen-antibody reactions is weaker in the case of the latter as compared to the original one. Example, M antigens are expressed by the bacteria Streptococcus pyogenes. These antigens elicit an immune response and produce antibodies against it. It is shown that these antibodies can cross-react with some of the skeletal muscle and myocardial proteins, leading to their damage. Precipitation reactions. A precipitate is formed on the interaction between the antibody and soluble antigen, whereas agglutination or clumping occurs during the interaction between the antibody and particular antigen. The precipitation reaction is dependent on the valency of antigen and antibody. The antibody should be bivalent, and the antigen can be bivalent or polyvalent. That is, the antigen can have either at least two copies of the same epitope or different epitopes. Depending on the antigen and antibody concentrations, three zones are revealed, as shown in Fig 4.114. The first zone, where the antibody concentration is high, therefore precipitation reaction is inhibited, and a small antigen-antibody complex is formed. Also, the unbounded antibody can be detected in the supernatant. There is another zone called an equivalent zone where the maximum precipitation of the antigen and antibody occurs, and large insoluble complexes of it are formed. Also, no antigen or antibody is detected in the supernatant. There is a third zone where due to the excessive antigen, precipitation is inhibited, and a small antigen-antibody complex is formed. Also, the unbounded antigen can be detected in the supernatant. Hence, the antigen-antibody ratio is linked to the size of its complex, which is further influenced by the concentration of antigen and antibody. Figure 4.114 precipitation reaction and formation of an immune complex. To a series of the tube 1 to 9, the same concentration of the antibody to the protein was added, followed by adding the increasing concentration of protein antigen in each tube, which resulted in the zone of antibody excess and small immune complex B, the zone of equivalence where all antigen and antibody forms large immune complex and are precipitated C, the zone of antigen excess with small immune complex. Generation of antibody diversity. Organization and expression of immunoglobulin multigenes. The vertebrate immune system is able to respond to the array of foreign antigens. Immunoglobulin study shows that they have a unique amino acid sequence in their variable region and a restricted number of invariant sequences in their constant region. This constancy of protein molecules and their ability to respond to a variety of foreign antigens is due to the organization of the immunoglobulin genes. The germline DNA non-B cell consists of a variety of gene segments which encode the heavy chain or light chain of the immunoglobulin, but these are only transcribed and translated when they are rearranged into the functional genes. There are 3 unlinked gene groups that are present on the different chromosomes and encode the immunoglobulin. 2 of them encode for the light chain, in which one encodes the kappa light chain and the other encodes the lambda light chain. The third gene group encodes for the heavy chain. There are many coding regions or exons in these gene groups which recombine at the DNA level to form a binding site. In the germline cell DNA, non-B cell, these exons are interrupted by the variety of non-coding DNA, whereas in DNA encoding T heavy chains and light chains, these exons are uninterrupted. The exons which encode the variable region consist of for heavy chain, B segment encoding 102 amino acids, D segment encoding 2 to 4 amino acids, and J segment encoding 14 or remaining amino acids. For light chain, B segment encoding 95 amino acids, and J segment encoding the remaining 13 amino acids. The exons which encode the constant region are separated from the variable region genes by J segment and non-coding DNA. Heavy chain gene group for the variable region. The first gene group which rearranges is the heavy chain gene group and is formed from 3 gene segments which are VH, DH, and JH. There are around 50 volts H genes, 20 DH genes, and 6 JH genes. One of the many D segment genes is moved to one of the many J segment genes, which creates the DJ combination. This DJ combination encodes the C-terminal part of the variable region of the heavy chain. The B segment is then moved to the DJ segment, which constructs the VDJ segment encoding the variable region of the complete heavy chain. Rearranged heavy chain consists of sequences in the given order: L-exon, intron, VDJ segment, intron, and various C segment genes. Among the C segment genes, micro and delta are closest to the VDJ segment. This rearranged DNA along with the C segment genes is transcribed to form the primary mRNA transcript. This primary mRNA transcript then undergoes alternative splicing to form either a VDJ micro or VDJ delta mRNA, as shown in Fig 4.115. For heavy chain. For each class and subclass, there is one functional C gene segment in each gene group. Therefore, the heavy chain gene group has 9 functional C gene segments, each encoding micro, delta, epsilon, alpha 1, alpha 2, gamma 1, gamma 2, gamma 3, gamma 4. For light chain. For the kappa light chain, there is one gene segment that encodes the kappa light chain. For the lambda light chain, there are 4 gene segments that encode the lambda light chain. Gene rearrangement. The gene segments described above will be selected by the single B cell and then will be translocated on the chromosome to create a DNA sequence which will encode the variable region or constant region. There are 2 recombination activating genes called RAG1 and RAG2, which are only expressed during the developing lymphocytes and help in breaking and rejoining the DNA during translocation and help in the generation of antibody diversity. Figure 4.115 heavy chain genes and their rearrangement. In a germline non-B cell, the heavy chain consists of many variable B segment genes, D segment genes and joining J segment genes. In developing B cell, one of the D segment first recombines with one of the J segments forming DJ combination, and then one of the B segment genes will recombine with the DJ combination, forming the VDJ gene. In each B cell, this rearranged gene will get transcribed, spliced and translated into the heavy chain protein. Light chain gene group for the variable region. The variable region of the light chain is encoded by the variable B segment and the joining J segment. The B segment gene encodes for the amino terminal residues, whereas the J segment gene encodes for carboxy terminal residues. Kappa light chain gene. There are 40 B kappa genes, 1 C kappa gene, and 5 J kappa genes present between the B kappa and C kappa gene lambda light chain gene. There are 30 B lambda genes and 4 C lambda genes each associated with the J lambda genes. After the rearrangement of the heavy chain, the B segment gene of either the kappa light chain or lambda light chain gene translocates to the J segment gene forming BJ recombination. In the case of the lambda light chain, as each J lambda segment gene is associated with the C lambda genes, translocation of AB gene segment to AJ gene segment results in AB region next to a particular C lambda gene. Rearranged light chain consists of sequences in the given order: L-exon, intron, BJ segment, intron, and constant region gene. Each B cell will use either the kappa light chain or lambda light chain. A primary mRNA transcript is formed from the BJ segment through the constant region of the light chain, which undergoes further splicing to remove the intervening non-coding sequences between the BJ segment and C region gene segment to form an mRNA for the complete light chain, as shown in Fig. 4.116. Figure 4.116 light chain genes and their rearrangement. During differentiation of the B cell and after the heavy chain genes are rearranged, one of the two following light chain groups rearrange. Either one of the B kappa gene segments of germline rearranges with the J gene segment forming the BJ combination, or B one of the B lambda genes of the germline recombines with one of the J segment associated with the C lambda gene to form BJ C lambda combination. The rearranged genes are then transcribed into the primary transcript, which undergoes splicing to form mRNA and is further translated to form light chain protein. Allelic exclusion. When the reading frame of the rearranged genes is correct and can be translated to produce a complete antibody, it is called productive rearrangement, whereas when the reading frame of the rearranged genes is not in the correct order, the gene encoding the functional protein is not generated and is called non-productive rearrangement. If one allele of the B cell rearranges non-productively, the other allele of it may productively rearrange. Normally, the gene products are derived from the genes of chromosomes of both parents. But, in B cell, the rearranged genes for heavy and light chains are from the chromosomes of one parent, either maternal or paternal, and not both. This is due to the process of allelic exclusion, which ensures the antigen specificity in which after the productive VH, DHJH rearrangement and VLJL rearrangement, the expression of other heavy and light chain genes is suppressed on the homologous chromosome, which ensures that the B cell is not multispecific. But, if the non-productive rearrangement occurs, the rearrangement process will continue and will not suppress the expression of other heavy and light chain genes until the productive rearrangement occurs. Therefore, the antibodies expressed on the surface of the B cells will be of the same specificity. Differential sequencing and class switching is mentioned above. Immature B cell first produces MIGM. On maturation, B cell then produces both MIGM and MIG, both of which are encoded by the same primary mRNA transcript, which undergoes differential cleavage and splicing, resulting in yielding either IgM heavy chain or IgD heavy chain. These are then translated and expressed along with the light chain on the surface of the B cell. Generally, a single class of immunoglobulin is expressed at one time by the B cell, but these cells are capable of switching to the other class of immunoglobulin, which is called class switching or isotype switching, which means they are able to switch the constant region of the heavy chain, micro or delta to alpha, epsilon, gamma, whereas the same variable region of the heavy chain continues to express. T helper cells stimulate the B cells by binding to the CD40 receptor present on the B cell with the CD40 ligand present on the T cells, which helps the B cells in the class switching. Also, class switching is also dependent on the cytokines that are synthesized by the T helper cells. Like, IL-4 induces the class switching to IgG, IL-5 switches it to IgA, IF and gamma to IG1. The above signals help in the class switching with the help of some switch regions or S regions present 5' to the constant regions of the heavy chain. The switch region is a repetitive intron sequence and is involved in the class switch recombination by translocating the BDJ coding region to associate with any of the CH genes. Recombination between the two S regions results in the excision of the sequences between them as a circular DNA molecule. There is an S micro region 5' to the C micro region, an S gamma 3 region 5' to the C gamma 3 region, and so on. For the C gamma 1 gene segment to express, which comes after the sequence C gamma 3 gene segment, there is a recombination between S micro and S gamma 1 regions due to which the sequences between the two are excised and help in the association of the BDJ segment and C gamma 1 segment and the class switching occurs. Ways of creating antibody diversity. Antibody diversity is the production of the antibodies having different specificities and ability to respond to the array of foreign antigens, which can be viewed to the various means. It segments. The one or two nucleotides of the B segment replaces all the three nucleotides of J segment, which can change the amino acid sequence at the point where the recombination occurs, which further results into the antibody diversity. After the class switching, the DNA of the variable regions of light and heavy chains become more prone to somatic mutations, which results in the change of nucleotide sequence and further the change in the amino acid sequence of the variable regions of the antibody expressed by the B cell. Consequently, the specificity of the B cell might change, or it may not be able to get stimulated by the original antigen. It might also happen that the attachment of antibody on B cell to the antigen is increased that will further contend with antigen more effectively than the initial B cell and will develop into the plasma cells, which will produce the antibodies of higher affinity called affinity maturation. Since these mutations take place at a larger rate, like 10 to 3 per base pair per generation, these are called somatic hypermutations. The other source of antibody diversity is that any of the 320 light chains can associate with any of the 8,262 heavy chain genes, which create a large number of the light chain and heavy chain combinations, thereby creating the unique binding sites for the antigen. Major histocompatibility complex (MHC) molecules. Major histocompatibility complex or MHC is a group of genes that are tightly linked and present in almost every vertebrate species. It helps in the intercellular organization and has the ability to distinguish between self and non-self. While antibodies respond to the antigens alone, the T cells are able to identify antigens only when they are bound to the MHC. Therefore, MHC acts as antigen-presenting cells. It is present in the humans on chromosome 6, referred to as HLA, human leukocyte antigen, and in mice on chromosome 17, referred as H2, histocompatibility 2. Many different forms or alleles of MHC exist at each locus. Therefore, it shows a high degree of polymorphism. Genetic. Can be antigen-independent mechanisms and antigen-dependent mechanisms which lead to antibody diversification. Antigen-independent mechanisms include Y multiple germline B, D and J of heavy chain genes and B and J of light chain genes at the DNA level. Y random combination of B, D and J of heavy chain genes and B and J of light chain genes at the DNA level. Y imprecise joining of B, D and J segments at the DNA level. Y random selection and pairing of different consolidation of variable regions of heavy and light chains in different B cells at the protein level. Antigen-dependent mechanisms include Y somatic mutation in the variable region at the DNA level increases the affinity of antibody binding sites. In the germline DNA of the human, there are approximately 51BH, 25D, 6JH, 40B kappa, 5J kappa, 31B lambda, and 4J lambda gene segments. Apart from this, it also consists of many pseudogenes. Therefore, multiple germline B, D and J segments lead to the diversification of the antigen binding sites of antibodies. In addition to this, in each developing B cell, the rearrangement of these gene segments is selected at random, thereby generating tons of B cells, each with different antigen specificity. For heavy chain diversity, the capability of any of the B segments to join with the D segment and J segment creates around 8,262 possible combinations for the heavy chain. 51 27 6 equals 8262. Similarly, for kappa light chain, there are around 200 possible combinations, 40 5, whereas for the lambda light chain, there are around 120 possible combinations, 31 4. These are the minimum calculations for generating the potential antibody diversity. Junctional flexibility further intensify the diversity during the recombination of B, D and J segments as there is an accurate ligation of these gene segments. Generally, during the translocation of the B segment to the J segment, the 3 nucleotides of the last codon of the B segment joins with the 3 nucleotides of the first codon of the J segment. But it is also possible that during the joining of these polymorphism is a single gene having multiple stable forms, and due to this, every individual has an uncommon cluster of hereditary MHC genes. Since the genes of MHC are tightly linked, these genes are inherited in groups from parents. Therefore, located on the same chromosome, within the MHC is present the cluster of the encoded alleles, which is referred to as haplotypes. One haplotype is inherited from both parents. MHC genes are classified into 3 types. Class 1 MHC genes, Class 2 MHC genes, Class 3 MHC genes. Class 1 MHC genes HLA-A, B, C. These genes encode glycoproteins which are expressed as 2 polypeptide chains on all nucleated cell surfaces. Out of the 2 polypeptide chains, one is a heavy chain or alpha, which is encoded by the MHC and is a transmembrane having a molecular weight of around 43 kDa. It has 3 extracellular domains, alpha 1, alpha 2, alpha 3. The second polypeptide chain called the light chain or beta 2 microglobulin, which exists on the surface of the nucleated cells, is encoded by a different chromosome that is chromosome 15. It has a molecular weight of around 12 kDa and is non-covalently attached to the class 1 MHC chain. Without the beta 2 microglobulin, the class 1 MHC chain is not able to express on the membrane of the nucleated cells. The level of expression of class 1 MHC molecules differs in different nucleated cells. Lymphocytes show high expression of these molecules, whereas liver hepatocytes, fibroblasts show lower levels of it. Also, it is absent in some nucleated cells like neurons and sperm cells. Target cells display peptides associated with MHCI molecule to the T cytotoxic cells. 3 independent genes encode class MHCI molecules or HLA-A, B, C. Class 2 MHC genes HLA-D. These genes encode glycoproteins which encode 2 chains alpha and beta and are found on antigen-presenting cells. The molecular weight of alpha and beta chains are 35 kDa and 28 kDa, respectively, and are expressed as transmembrane glycoproteins with the extracellular domains alpha 1, alpha 2, beta 1, beta 2. Antigen-presenting cells are of 2 types, professional and non-professional. The former are the ones which include B cells, macrophages, dendritic cells and express class MHC2 genes, whereas the latter include fibroblasts, glial cells, which can be induced to express class MHC2 genes. Therefore, the majority of the cells express only MHCI molecules in the absence of the inducing factors. Some cells express both like B cells. Antigen-presenting cells display peptides associated with MHC2 molecule to the T helper cells. 3 sets of HLA-D gene encode class MHC2 molecules or HLA-DP, HLA-DQ, HLA drive. Figure 4.117 class 1 and class 2 MHC molecules having external domains, transmembrane segment and cytoplasmic tail. Class 3 MHC genes. These genes encode many secreted proteins which have immune functions, such as components of the complement system and molecule, which are involved in inflammation or heat shock proteins. Class 1 and class 2 MHC molecules encode both classical MHC molecules as well as non-classical MHC molecules. Classical MHC molecules are the ones which are peptide antigen-presenting MHC molecules, whereas non-classical MHC molecules are those which do not play a role in the peptide presentation to T cells. On the other hand, class 3 MHC molecules play no role in both types of these MHC molecules. Antigen processing and presentation. There is a peptide binding cleft in both class 1 and class 2 MHC molecules. But both the ends in the cleft of class 1 MHC is blocked due to which peptides containing 8 to 10 amino acid residues can only bind, whereas the cleft of the class MHC2 is open due to which slightly longer peptides of 13 to 18 amino acid residues can bind. Also, the peptide should contain specific amino acid residues near N and C terminals for binding to the class 1 MHC molecules, whereas there is no such necessity for class 2 MHC molecules. Both types of T cells, T cytotoxic and T helper cells, are only able to recognize the antigens which are bound to the MHC molecules, and this is called self-MHC restriction. CD8-TC or cytotoxic cells are able to recognize antigens which are bound to the class 1 MHC molecules on target cells, whereas CD4-TH or T helper cells are able to recognize the antigens bound to the class 2 MHC molecules. Endogenous antigens generated within the body cells. Antigens use the cytosolic pathway to present the antigen to the cytotoxic T cells, whereas exogenous antigens which come from outside use the endocytic pathway to present the antigen to the T helper cells. Class 1 processing pathway, cytosolic pathway. The bacteria, viruses and parasites that infect the host cells are referred to as endogenous antigens. The processing of these endogenous antigens takes place in the cytoplasm and hence referred as cytosolic pathway. Firstly, the huge protein complex called proteasome cleaves the proteins into peptides in the cytoplasm. There are some other cytosolic enzymes like aminopeptidase which further degrades these peptides. These peptides are then transported into the endoplasmic reticulum with the help of specific transporter proteins called transporter associated with antigen processing tap. On reaching this intracellular compartment, these peptides attach to the class 1 MHC molecule, which is present near the tap and is held at that site with the help of chaperone proteins which include calix and calreticulin and so on. The peptide MHCI complex is then free from the chaperone and is exported to the cell surface, where it fuses with the membrane, migrates to the surface and can be seen by the cytotoxic T cells having CD8. Class 2 processing pathway, endocytic pathway. Unlike viruses and some bacteria whose replication occurs in the cytosol, there are some pathogens like mycobacteria, leishmania whose replication occurs in cellular vesicles. The foreign exogenous protein is endocytosed in the endosomes and degrades into the peptides due to the acidic environment. Class 2 MHC molecules with the invariant chain to prevent the premature binding of peptides are present in the endoplasmic reticulum, which are transported to the late endosome. The invariant chain is cleaved by the proteases, but a small part of it remains attached, which is called clip for class 2 associated invariant chain peptide to prevent the premature binding of peptides. On fusion with the endocytic vesicles, the clip is released and the peptide binds to the class 2 MHC molecule, which is moved to the plasma membrane, where it will be identified by the CD4 T helper cells. Figure 4.118 comparison of the antigen processing pathway by class 1 MHC, cytosolic pathway, and class 2 MHC, endocytic pathway. B cell maturation and activation. B cell maturation. The synthesis and maturation of the B cells take place in the embryo first and continues throughout life. Before birth, the B cell maturation occurs in the yolk sac, fetal liver, fetal bone marrow. After birth, the major site for B cell maturation is bone marrow. Lymphoid stem cells develop into progenitor B cell, pro B cell, in the bone marrow, which further develop into precursor B cell, pre B cell. It has been shown that the microenvironment of the bone marrow stromal cells is crucial for this differentiation process, as proved by isolating pro B cells from the bone marrow, which were then cultured in vitro in the absence of the stromal cells failed to progress into pre B cell stage. It was due to the fact that the stromal cells help in the development process by secreting cytokines like IL-17 and by mediating the interactions with the pro B cell and pre B cell with the help of some adhesion molecules, thereby supporting the developmental process. During the pro B cell stage, the variable region of the heavy chain is rearranged VDJ rearrangement. As discussed earlier, since the rearrangement takes place in a million ways in these developing cells, diversification of antibodies occurs, each antibody with a different specificity. This diversification takes place when foreign protein is not present and produces a large number of mature B cell in which some have specificity against the foreign particles. Together with the variable region of the heavy chain, the constant region of the heavy chain, in particular, micro-heavy chain is transcribed and differentiated into pre-B cell. In the pre-B cell, the micro-heavy chain gets associated with the surrogate light chain, which is a complex of 2 proteins associated non-covalently, BPREBB-like sequence and lambda-5C-like sequence, and forms a light chain-like structure. These are called surrogate light chains are called invariant as they are synthesized in pre-B cells and are identical in all B cells. At this stage, the light chain gene will rearrange. If productive rearrangement occurs, the heavy and light chains will combine and will give rise to the immature B cell, which will express functional membrane IgM. This IgM, along with the Ig alpha and Ig beta, which are the transmembrane molecules, will form the pre-B cell receptor. After the formation of the B cell receptor, many cell divisions of each pre-B cell will take place, which will result in many progeny pre-B cells, which will rearrange with different light chain gene segments leading to antibody diversification. It has been shown that the pre-B cell receptor is important for it to continue to the immature B cell stage as the signal is generated through this receptor. Further development will produce naive B cells expressing vitamin MIG, which will leave the bone marrow, circulate in the blood and will enter the secondary lymphoid organs like the spleen or lymph node, where these cells will encounter antigens and will become mature B cell. The mature B cell, on encountering the antigen, becomes activated and will further undergo the clonal proliferation in which the majority of these cells will be transformed into the plasma cells, which are the antibody-secreting cells, whereas some activated cells will become long-lived memory cells. Both membrane-bound and secreted forms can exist for all the classes of the antibody. The difference between the two forms is due to the carboxyl terminus of the heavy chain. B cell receptor signaling. As mentioned above, B cell receptor is a complex which consists of IgM, which binds to the antigen, associated with the Ig alpha and Ig beta, which are the signal transducing transmembrane molecules. There are other B cell co-receptors which affect the signal transmitted through this receptor. These include CD19, CD81 and CD21. In the cytoplasmic tails of Ig alpha, Ig beta, there are present ITAMs, immunoreceptor tyrosinase activation motifs, the tyrosines of which get phosphorylated on the binding of antigen with the IgM. These tyrosines and ITAMs are linked to the SRC family tyrosine kinase and are able to get phosphorylated with the help of it. These phosphorylated tyrosines then bind to the SYK spleen tyrosine kinase, which helps in the formation of signalosome, which consists of B cell receptor, SRC family tyrosine kinases, adapter proteins and signaling enzymes which help in the transduction of the signals. Figure 4.119 The B cell receptor. B cell receptors are complexes consisting of various components, a membrane-tethered IgM antibody and associated membrane-bound protein CD79A and CD79B. B cell activation. Types of B lymphocytes. Based on whether the B cells need T cell help for proliferation and differentiation, they are of two types, B1 cells and B2 cells. Antigens are TD antigens. TH cells induce the B cells to proliferate, differentiate and synthesize antibodies. Also, they induce the class switching of the antibodies and affinity maturation. This requires the direct contact of TH cells with the B cells. The humoral response to these thymus-dependent antigens is stronger than the response to thymus-dependent antigens and involves affinity maturation, class switching and immunological memory. Activation of B cells with the help of TH cells. Three signals are required for the activation of B cells by the soluble antigens dependent of TH cells. Firstly, the thymus-dependent antigen binds to the antigen-specific membrane antibodies present on the surface of the B cells. This cross-linking helps in generating the signal 1, which helps in the increased expression of various cell membrane molecules of B cells, including class 2 MHC molecules and co-stimulatory ligand B7. The expression of these molecules makes the B cell an antigen-presenting cell or APCs and differs from other APCs by binding specifically to the antigen. Once the antigen binds to the antibody of B cells and generates signal 1, it is internalized by the receptor-mediated endocytosis and is degraded into peptides by undergoing the endocytic pathway. Some of these peptides then bind to the class 2 MHC molecules and are presented on the surface of the B cells to the TH cells. The TH cell then recognizes this processed antigenic peptide displayed by the class 2 MHC molecule present on the surface of the B cell, and these two cells then further interact to form TB conjugate. This conjugate helps in upregulating the expression of CD40 ligand on TH cell surface. This CD40 ligand on TH cell binds to the CD40 receptor on the B cell, thereby generating the signal 2 and activates the B cell. Further co-stimulatory signals between B7 of B cell and CD28 of TH cell stimulate both B cell and TH cell. The stimulated TH cell then sends signal 3, which includes the release of cytokines like IL-2, IL-4, IL-5 that bind to the receptors on the B cell and help in their activation. B1 cells are derived from the stem cells in the bone marrow during fetal life and synthesize mainly surface IgM antibodies having lower affinity. They can recognize multiple, repeating antigenic determinants and do not require T cell help for proliferation and differentiation and are therefore T cell independent. The response of B1 cells to protein antigens is poor and has little or no immunological memory. B2 cells are called conventional B cells as they are mainly involved in the development of humoral immune responses. They are developed in the bone marrow and require T cell help for proliferation and differentiation and are therefore T cell dependent and result in memory B cells and antibody secreting plasma cells that produce IgG, and IgG, which have higher affinity. The response of B2 cells to the protein antigens is high and has immunological memory. TH cells independent pathway. Some B cells do not need the help of T cells for their activation and follow TH cells independent pathway. Antigens such as thymus-independent antigens or T1 antigens help in activating the B cells without the direct involvement of TH cells. T1 antigens are of two types, type 1 antigens and type 2 antigens. Type 1 antigens include the bacterial polysaccharides which present in high concentrations have the ability to activate B cells independent of their specific antigen receptors, whereas type 2 antigens include linear antigens which are not easily debased and have epitope spaced appropriately on the molecule like pneumococcus polysaccharide. These are present on the surface of the splenic marginal zone and help in activating the B cells through cross-linking of their surface receptors. The humoral response to these thymus-independent antigens is weaker than the response to thymus-dependent antigens and does not involve affinity maturation, class switching and immunological memory. TH cells dependent pathway. Most of the B cells, in cooperation with the TH cells, respond to the thymus-dependent B cells. After activation, move to the specialized regions of lymph node and spleen for the differentiation process resulting in the production of antibody-secreting plasma cells and memory B cells. Some of the antigen-stimulated B cells move to the follicles of lymph nodes and spleen, where they divide and differentiate and are developed into the germinal center, which consists of proliferating B cells. Within these germinal centers, the Ig genes undergo class switching and somatic hypermutation depending on the activity of the germinal center enzyme. Activation-induced cytidine deaminase 8. Figure 4.120 Activation of B cell by the TH cells. When the multivalent antigen binds to the MYC present on the B cell, signal 1 is generated. The activated T cell binds to the B cell through the antigen receptor as well as through the interaction between CD40 on B cell and CD40L, CD154 on the activated TH cell to generate the signal 2. The activation process is completed by delivering cytokines by the bound T cell. Clonal selection theory. Clonal selection theory explains how the highly specific adaptive immune system responds against the highly diverse antigens. According to this theory, the host first generates a variety of lymphocytes having different specificity. When the foreign antigen is induced, very few B cells will have receptors specific to this antigen. Therefore, one of these B cell will then undergo the process of antigen processing and presentation and further the activation process with the help of the TH cell, which will be specific to the peptide generated from that antigen. After the activation and proliferation of the B cell, clones of a large number of daughter cells are produced. Some of them will develop into memory cells, whereas others will differentiate and will form plasma cells which will secrete the specific antibody. Also, the direct T cell interaction with the B cell will help in inducing the class switching depending on the type of helper cell and the cytokines it secretes. T cell maturation and activation. T cells are involved in dealing with intracellular pathogens like viruses, bacteria and in helping the B cell responses. The generation and selection of T cells occur in the thymus. T helper cells are able to recognize the processed antigens bound to MHC class 2 molecules, whereas T cytotoxic cells are able to recognize the processed antigens bound to MHC class 1 molecules. These cells are able to recognize the processes of antigens bound to the MHC molecules with the help of a specific T cell antigen receptor or TCR. This recognition by TCR helps in generating the signals, which leads to the transcription of genes that encode cytokines that help in the activation of the T cells. T cell receptor TCR for antigen. Like B cells, T cells express TCR, which is antigen specific, but unlike B cell receptors or Ig, the TCR is only expressed on the T cell membrane and not in the secretion such as these cells have much broader specificity for the recognition of the antigens and do not require it to be bound to the MHC molecules. TCR complex. The antigen receptor, either Ab or Grand dimers associated non-covalently to the CD3 molecule, forms the TCR complex. The CD3 molecule is made up of 5 polypeptide chains which are associated to form 3 types of dimers: gay heterodimer, dihedrodimer, and X-homodimer or Z-heterodimer. These polypeptides help in the T cell signaling and are activated only when the antigen binds to the TCR. CD3 acts as a chaperone and is the same on all T cells. Co-receptor molecules are the transmembrane molecules which are present in association with the TCR. It can be either CD4 on mature T cell or CD8 on mature T cell. Both CD4 and CD8 attach to the non-polymorphic region of MHC molecules. CD4 attaches to MHC class 2 molecules, whereas CD8 attaches to MHC class 1 molecules. Both CD4 and CD8 help in the transduction of the signal after the antigen binds to the TCR. The intracellular portions of both CD4 and CD8 are linked to the tyrosine kinase enzymes, which help in the T cell activation pathways. Serum. There are 2 types of T cell receptors based on the transmembrane glycoprotein chains AbTCR or Grand TCR, both of which develop in the thymus. These glycosylated chains have variable and constant domains. Both the chains, either Ab or Grand, are linked with the help of interchain disulfide bond. Alpha, beta, or AbTCR. Most of the human peripheral T cells express AbTCR, and their maturation takes place in the secondary lymphoid organs. T helper and T cytotoxic cells are the Ab T cells which are involved in the protection against the invading pathogens as well as provide help for the humoral response. The alpha chain has a molecular weight of about 50 kDa, whereas the beta chain has a molecular weight of about around 39 kDa. The variable regions of both the chains form the T-cell antigen binding site, which can only recognize the processed antigens bound to the MHC molecules. Gamma delta or GG delta G delta TCR. Very low numbers of human peripheral T cells express GRAN TCR, but their number increases during infection. The morphology of GRAN T cells is similar to natural killer cells, unlike GB T cells, which interact only with the processed antigen. Figure 4.121 The T cell receptor. Cells. The negative thymic selection includes the selection of those cells whose TCRs have a weak affinity to the self-MHC. Most of the cells, after the selection, become have TCR CD4+ or CD8+. Cells, which is dependent on the signal continuity these DP thymocytes through their TCR. Some develop into other lineages, which include GRAN TCR CD4+ or CD8+ cells, natural killer T cells or dendritic cells, each having a different function. The T cells which express TCR specific to self and foreign antigens develop in the thymus and undergo positive selection. After the positive selection, the T cells become self-MHC restricted, that is, they are only able to recognize the peptide bound to the self-MHC and not foreign MHC proteins. Negative selection ensures the death of those T cells which have strong reactivity towards the self-antigens as it can lead to various autoimmune diseases otherwise. Therefore, the ones having the weak binding to the self-antigens are able to survive after the negative selection, which results in developing self-tolerance. The developing T cells are educated in the thymus to distinguish between self and non-self. The ability to be not able to react with the self cells is called self-tolerance, which is acquired during the T cell maturation. The T cell precursors are subjected to the positive and negative selection in the thymus, which ensures the survival and maturation of only those T cells that can recognize peptides bound to the self-MHC but are not autoreactive, that is, are not able to react with self-antigens. If there is an error in any of the developmental processes, including not able to rearrange the TCR, failure in the negative selection or in the positive selection, it results in the apoptosis of those cells and therefore, due to this tight selection, approximately 95% of the T cells die in the thymus before they are ready to mature and move to the peripheral lymphoid organs. Schematic diagram of TCR engagement with the peptide antigen MHC complex using the CD4 T cell as an example. The TCR complex is made of the antigen receptor, either AB dimer or GRAN dimer, which are attached non-covalently to CD3 molecule that forms three types of the dimer: gay heterodimer, deheterodimer, and X-homodimer or Z heterodimer that helps in the T cell signaling. Co-receptor molecules such as CD4 or CD8 are also associated with TCR. T cell maturation and thymic selection. Thymus is the site for T cell maturation. The epithelial cells of the thymus secrete various chemotactic factors, which help in the migration of progenitor or pro-T cells from the site of hematopoiesis, that is bone marrow, into the thymus. These precursor T cells start developing, which are called thymocytes and are subjected to proliferation and differentiation to form subpopulations mature T cells that are functionally distinct. The immature thymocytes are double negative cells, DN cells, or CD4 CD8 as they do not express either of the co-receptors CD4 or CD8. These DN cells then undergo four stages of development DN1 DN4 to form the double positive cells DP cells, or CD4 plus CD8 plus based on the presence and absence of cell surface molecules, c-KIT, CD44 and CD25. The T cell receptor TCR gene loci are rearranged in this developmental process. First, these double negative T cells rearrange their TCR beta chain and those cells which are successful in this step then begin the rearrangement of the TCR alpha chain. They now become double positive thymocytes DP cells, or CD4 plus CD8 plus. These DP thymocytes are then subjected to the positive and negative thymic selection in the thymus cortex. The positive thymic selection includes the selection of those cells whose TCRs respond to the self-MHC, which are expressed by the thymic cortical epithelial. Figure 4.122 Process of thymic selection. The T cell precursors are formed from lymphoid stem cells. These precursors, after entering the thymus, undergo multiple gene rearrangements to develop the T cell antigen receptors and CD4 or CD8 molecules and further the positive and negative selection. These then escape the thymus as CD4 plus T helper cells or CD8 plus T cytotoxic cells and move to the secondary lymphoid organs. T cell activation. After the thymic selection, these naive T cells move to the secondary lymphoid organs for their activation, which results in the generation of effector cells and memory T cells. These cells are called naive because they are mature but have not encountered the antigen. Three kinds of signals are needed for the activation of naive T cells. Antigen-presenting cells or APCs are present in the secondary lymphoid organs, which help in the antigen processing and presentation. Signal 1 is generated when the processed antigen bound to the MHC molecule on the APCs is recognized by the T cell through the T cell receptor. There are some accessory molecules, which strengthen the binding of T cell and APC, which includes adhesion molecules like integrins, ICAMs, intercellular adhesion molecules on APC and LFA, lymphocyte function-associated antigens on T cell. The other signal which is also required for the clonal expansion is signal 2. Signal 2 is generated by the co-stimulatory molecules, which include B7 on APC that binds to the CD28 on the T cell. The T cell receiving only signal 1 but no signal 2 will result in energy or will be turned off. Signal 3 is generated in the form of cytokines, which determines the type of responder the cell will become. Like, if the cells are exposed to IL-12, they will become Th1. If they are exposed to IL-4, they will become Th2 and so on. All three signals help in the clonal expansion and differentiation of the T cell, which results in the generation of effector helper or cytotoxic T cells. Almost all cells in the host express class 1 MHC molecules, which are recognized by the CD8 plus T cells and become the cytotoxic T cells having the ability to kill the infected cells. The APCs express class 2 MHC molecules, which are recognized by the CD4 plus T cells and become helper T cells and have the ability to secrete cytokines which positively or negatively regulate other cells like B cells, macrophages or other T cells. Figure 4.123 Activation of the T cell. Signal 1, which is generated when the T cell recognizes the processed antigen bound to the MHC on APC and co-stimulatory signal 2 between B7 on APC and CD28 on T cell, helps in upregulating the expression of IL-2 and its receptor, signal 3, thereby allowing the T cell to proliferate. Immunological synapse is the region which is formed between TCR accessory and co-receptor molecules present on the T cell surface and ligands on the APC. The molecules present on the T cell surface help in conveying the signal from the T cell surface to the nuclei via cytosol, helping in the gene transcription. A naive helper T cell on activation in a peripheral lymphoid tissue can differentiate into different subpopulations of effector cells: Th1, Th2, Th17, TFH, and ITREG. Th2 and TFH help in augmenting the humoral immune response via the activation of B cells. Th1 and Th17 help in augmenting the cell-mediated immune response, whereas ITREG helps in inhibiting the T cell responses. Apart from the effector cells, which are short-lived, long-lived memory T cells are also produced, which provide protection against secondary infection. Activation by superantigens. There are some protein products of bacteria or viruses which are able to bind simultaneously to the variable region of the beta chain of the TCR and to the alpha chain of the class 2 MHC molecule, as shown in Fig 4.124. The superantigens do not undergo the process of antigen processing and presentation. Instead, having the ability to bind to a specific family of TCR, they are able to bind to the APC while simultaneously stimulating the T cell. As a result, irrespective of the antigen specificity, a large number of T cells is activated, which produces a large number of cytokines. Examples include staphylococcal enterotoxins and toxic shock syndrome toxins, which are the bacterial superantigens. Figure 4.124 Activation of superantigens. Superantigens help in the activation of the Th cell by binding to the variable region of the beta chain of the TCR and to the alpha chain of the class 2 MHC molecule. Cell-mediated immunity. The naive cytotoxic T cell, on activation, develops into effector cells called cytotoxic T lymphocytes, CTLs, which help in killing the virus or bacteria-infected cells, tumor cells. These cells also release cytokines that help in the killing of the target cells like TNF-beta. The mature CTLs help in the apoptosis of virus-infected cells or other cells by two pathways: perforin-induced apoptosis, large cytotoxic granules which contain proteases, granzymes, and perforin are present in the CTL. On the establishment of the conjugate between CTL and the target cell, the granules of the CTL migrate to the point of the conjugation where the content of the granules is released into the target cell. Perforins polymerize the target cell membrane, which is a Ca plus 2 dependent process creating pores in the membrane, thereby allowing the entry of proteases into the cell. These proteases then cleave the cellular protein and result in the apoptosis of the target cell. FOS-mediated apoptosis. There is an increased expression of the FOS molecule on the surface of the virus-infected cells, which interacts with the FOS ligand present on the CTL. Due to this interaction, the death domain of the FOS molecule becomes activated and then binds to the adaptor protein. As a result, specific clustered procaspases are recruited that cleave each other to form active caspase molecules that are responsible for initiating the caspase cascade resulting in apoptosis. Immunization. The experiments done by the Lady Mary Wortley Montagu in 1718 and Jenner's work in 1796 led to the foundation of immunization and vaccines. In the 19th century, experiments by Louis Pasteur and Emile Roux demonstrated that protection by an activated or attenuated Pasteur in honor of Jenner's work with cowpox inoculation. Although the above experiments had shown success in providing immunity against life-threatening diseases and underlying pathogens and had led to the development of a number of vaccines available today, the mechanism of organisms could provide immunity against disease. The attenuated strain is named vaccine by how this immunity was produced remained unknown till longer duration. The term variolation differs from vaccination, in which dried pustules of smallpox caused by the variola virus from a sick individual were inoculated into a healthy individual to prevent the healthy individual from developing the disease. It was developed in Central Asia in the 2nd millennium and practiced, which was then spread east to China and west to Turkey, Africa, and Europe. Immunization is the process in which a person is fortified immunity or becomes resistant against a disease-causing pathogen known as an immunogen and thus provides a long-lived done using live, attenuated vaccines, which produce a complex immunologic, both humoral and cell-mediated response to the natural infection. The immunity introduced by the live vaccines can be subdued by the antibodies, received passively either from the mother by transplacental acquisition or by receiving the blood-containing immunoglobulins, and the decline in passive antibodies level results in the optimum immune response. For example, vaccination for measles at the age of 12 months of age when antibody level acquired from the mother decreases, the immune response could be optimum. Generally, after the administration of a single dose, the immune response may be only 90–95%. Therefore, to boost immunity in a community and to prevent the spreading of disease, multiple dose regimen is required. An excellent example of natural active immunization is exposure to influenza. The body begins the process of developing long-term immunity to the influenza virus. The effectiveness of the live attenuated vaccines labs with decreased pathogenicity depends on how the immune system replicates and elicits a response to natural infection. It is usually effective with a single dose. The labs are available for diseases such as measles, mumps, rubella, MMR, rotavirus, varicella, yellow fever, and influenza LAIV. The artificial active immunization procedure includes the preparation of either the whole microbe that is inactivated or its parts that are purified, which acts as an antigen to induce an immune response. The immunizations done in this manner do not result in active disease as the microbe is treated or made inactive before using it as an antigen so that it should have any harmful effect on an infected person. Moreover, it would mimic the stimulation necessary for immune development. State of protective immunity against infectious disease. Immunization is used to control or sometimes eliminate the infectious diseases that are responsible for the 2 and 3 million deaths each year. Immunization is considered a cost-effective health intervention. Despite being controversial as claimed to contaminate or unnatural, immunization is most widespread and accessible to even the most hard-to-reach and vulnerable populations and can be delivered effectively through outreach activities. Immunization helps to develop children's immune systems as well as protects children from the deadly diseases. Some of the diseases, for example, polio, have almost completely been eradicated throughout world through immunization by scheduled vaccination. Immunization can be achieved in 2 ways. 1. Active means. 2. Passive means. Concept of active immunization. Active immunization occurs when an individual is exposed to or comes in contact with microbes leading to the generation of antibodies against the microbe by inducing an adaptive immune response which takes days to weeks to develop and is usually long-lasting, possibly lifelong. In active immunization, as the name implies, the immune system plays an active role. The proliferation of antigen-reactive T and B cells is induced and results in the formation of protective memory cells. The immune response occurs efficiently upon the next encounter with this same microbe and can be very efficient, as seen in many childhood infections in which an individual gets immune after the first exposure to the pathogenic microbe. It is achieved by using 2 major approaches, either naturally or artificially. The natural active immunization recommended program by health authorities in many countries. However, vaccination is not always 100% effective. Moreover, in some cases, for example, measles, the estimated immunity threshold reaches approximately 92–94%, falling below the 95% threshold is set by WHO. With any vaccine, a small percentage of recipients will respond poorly and therefore will not be adequately protected. However, if the majority of a population, i.e., a community is protected by vaccination and thus making them immune to an infectious agent resulting in reducing the pathogen reservoir significantly. In that case, even individuals who are not vaccinated as well as those who fail to make an adequate immune response to the vaccine can be indirectly protected as the chance of a susceptible individual having exposure of an infected individual remains very low. This observable fact is called herd immunity. The decrease in vaccinations results in lowering herd immunity and the appearance of disease epidemics. In the mid-1990s, a diphtheria epidemic raged due to a decrease in herd immunity resulting from decreased vaccination rates in many regions of new countries formed by the division of the Soviet Union. Similarly, in the United States, measles epidemics reappeared among college students and preschool-aged children because of an overall decrease in vaccinations, which resulted in lowering the herd immunity of the population. Therefore, for the individual as well as community protection against diseases, and to make vaccination more effective, high levels of coverage for making immune a population via vaccines is important. This will not only be required for individual protection but also prevents diseases in vulnerable populations that are protected by vaccination. This will result in achieving high population immunity or herd immunity, such as removing barriers that may prevent access to vaccines, e.g., providing recommended vaccines without cost, as well as mandates for immunization requirements for attending school. This prompted the revised recommendation that children receive 2 MMR immunizations, one at 12 to 15 months of age and the second at 4 to 6 years. Figure 4.125 Difference between natural and artificial active immunity. Natural immunity is acquired by the infection with any pathogenic organism, whereas artificial immunity is provided to an individual via vaccination. Thus, the goal of active immunization is to trigger the adaptive immune response in a way that will elicit protective immunity and immunologic memory. Active immunization with various types of vaccines has played an important role in the reduction of deaths from infectious diseases, especially among children. Concept of vaccination. Vaccination is a procedure in which an intentional exposure in the form of antigenic material, vaccine, that does not cause disease is made to stimulate an individual immune system to develop an adaptive immunity against it. Ideally, exposure to antigen would result in the formation of effectors lymphocytes that help to eliminate the pathogen and generation of memory cells, thereby providing long-life protection against that pathogen. The development of memory B or T cells specific to the pathogen, i.e. development of a protective memory response is a potential outcome of vaccination. Over the past 100 years, vaccination has been proved to be one of the most effective public health interventions in saving lives against many infectious diseases and is the most. Figure 4.126 A comparative analysis of the vaccination in the case of community protection. A community is presented with poor coverage of vaccination where very few people are vaccinated. In this case, the population is highly susceptible to infection, where if a person capable of transmitting a disease comes in contact with a susceptible person, it will lead to transmission of an infection, forming a chain resulting in the outbreak of disease. B. A population that is highly immune due to good coverage of vaccination. Here, if a person capable of transmitting a disease comes in contact with a susceptible person, it will break the chain of transmission since the person is not exposed. Concept of passive immunization. Passive immunization occurs via the transfer of antibodies from a donor to an unimmunized individual known as the recipient. This results in the development of a temporary immunity against a particular organism or toxin. The administration of the preformed antibodies, raised against a disease, of a donor via the passive immunization prevents the occurrence of disease through the neutralization of the invading microorganism by various approaches such as 1. By directly preventing their entry into cells. 2. Blocking cell surface receptors, therefore preventing microbes' entry into cells. 3. By activating the complement cascade eliminates the pathogen. 4. By the process of opsonization, i.e., coating the pathogen to assist in phagocytosis by immune cells. 5. Antibody-dependent or complement-dependent destruction of infected cells. The immunity against a disease lasts till the injected antibodies are present in the recipient's extracellular spaces, and once antibodies are destroyed, the individual would no longer have immunity against a given microorganism or toxin. That means passive immunity provides immediate protection against infection, and since it will not induce to develop a memory, which is induced in active immunity, therefore, the individual is always at risk of getting infected by the same pathogen later. Chronic B-cell lymphocytic leukemia, primary immunodeficiencies, hypogammaglobulinemia, immune thrombocytopenia, and prevention of graft versus host disease. These antibodies are used. Side effects of IVIG that could occur such as fever, chills, headache, faintness, nausea, vomiting, hypersensitivity, anaphylactic reactions, and coughing. Subcutaneous immunoglobulin (SCIG). SCIG is used for the treatment of individuals with primary immunodeficiency and is administered subcutaneously. It is also prepared from pooled plasma collected from thousands of donors. Further, it was treated to remove contaminants to ensure product safety and integrity. The side effects, such as fever, chills, etc., with SCIG injections, are less common than with IVIG. Hyperimmune globulin. Hyperimmune globulin is prepared from the plasma of the donor convalesced from natural infections or artificially immunized, having high titers of antibodies against a specific antigen. Hyperimmune globulins are available for hepatitis B, infant botulism, rabies, tetanus, cytomegalovirus, vaccinia, and varicella zoster. The administration of these antibodies is painful, and sometimes it may lead to anaphylaxis. Monoclonal antibodies. Monoclonal antibodies specifically active against pathogens or infectious agents are of great therapeutic value and are currently an interesting topic of study. Tocilizumab was the first mAb used and licensed against the respiratory syncytial virus (RSV) for the prevention of infection in high-risk infants. Thus, the currently active areas of monoclonal development have focused on viral diseases without available vaccines, e.g. HIV, Ebola, SARS, MERS, viral diseases with limited effective antiviral drugs, e.g. influenza, rabies, and bacterial toxin-mediated diseases, e.g. anthrax, C. difficile colitis. The monoclonal antibody products currently licensed or progressing towards late-stage development for infectious diseases are listed in tab 4.30. Passive immunization also occurs via two means, i.e., either naturally or artificially. Natural passive immunization occurs via the passage of maternal antibodies to fetuses and infants through the placenta and colostrum and milk, respectively. Artificial passive immunization is induced via the administration of antibodies obtained from humans, horses, or other animals specific to a pathogen or toxin to a non-immune individual. These administered antibodies are used in several forms, such as human or animal blood plasma or serum, as pooled human immunoglobulin for intravenous IVIG or intramuscular IG, and as monoclonal antibodies mAb. Human immunoglobulin IG. IG is a concentrated solution of antibodies, consisting of mainly Ig and trace amounts of IgA, IgM, along with other serum proteins. This concentrated solution of IG is derived from the plasma proteins of the donor. IG, if stored at 4 degrees Celsius, has high stability, which lasts for months, having half-life in circulation is about 3 weeks, and it rarely contains any transmissible viruses such as hepatitis B or C, HIV. IG is administered intramuscular IM. IG must be administered soon after exposure to antigen because after 48 h of IM injection, serum antibody levels rises to the maximum. IG may be used for prophylaxis in diseases such as Y hepatitis A, Y measles, Y immunoglobulin deficiency, Y varicella zoster IG, in immunocompromised patients. Varicella is used Y rubella, exposure during the first trimester of pregnancy. IV immunoglobulin, IVIG. IVIG was developed to provide larger and repeated doses of human immunoglobulin IG and is used to treat severe infections caused by bacteria and viruses. For the autoimmune and immunodeficiency disorders, for example, Kawasaki disease, HIV infection in children, S number. Name of the diseases. Monoclonal. Antibodies mAb descriptions. Human mAb status indications. 1. Anthrax Anthran Raxibacumab. To anthrax toxin license 2012. Like adaptive and innate immunity, stress responses, inflammation along with cellular growth and apoptotic responses. Why Notch signaling pathways play a very crucial role in the transmission of information associated with development like embryogenesis. Notch signaling pathway affects cellular differentiation in a wide spectrum of the organism and has a unique feature of a very simple structure and very few core components. Why multifunctional transcriptional factor P53 regulates more than 2000 gene activity. P53 has major impact on maintaining genomic stability along with fidelity, many cellular processes, metabolism, cell longevity, and is a tumor suppressor. It gets activated as a stress response which could cause genotoxic stress, hypoxia, oncogenic trauma, and heat shock. It is associated with more than 50% of cancers in humans, and hence has been a very promising and potent biological target in cancer drug development. Why the ROS-RAF-MEK-ERK signaling pathway is a typical MAPK signaling pathway implicated in external stimulus-mediated cell growth, differentiation, and cellular death. The external stimulus could be a growth factor, inflammatory cytokines, or could be environmental stress. This cascade of signals combines signals from cell surface receptors to regulate gene expression by acting on transcription factors. Depending on the type of stimulus obtained from cell surface receptors, the fate of downstream signaling is decided, which could. Why once phosphorylated, AKT regulates various cellular functions downstream like cell survival, proliferation, metabolism, cellular growth, and many others by either activating or inhibiting molecules like FOXOs, BAD, GSK3, MDM2, TSC2, caspase 9, AS160, PROS40, and ENOS by phosphorylating. Why proto-oncogene receptor tyrosine kinase c-MET and its ligand hepatocyte growth factor HGF have been reported to be associated with various types of malignancies. HGF secreted by mesenchymal cells has a pleiotropic effect as well as cytokine and influences cellular function by promoting cell survival, proliferation, scattering, motility, and morphogenesis. It also plays a very important protective function in various disease conditions like liver cirrhosis, progressive neuropathies, and so on. HER2 is a human analog of the oncogene NOI in rodents. This gene is homologous to the epidermal growth factor receptor EGFR gene. It has been observed that EGFR-related gene gets intensified in the human breast cancer cell line and is known as HER2. HER2 family proteins are associated with various cellular processes like cell survival, proliferation, motility, cell cycle regulation, and so on by interacting with downstream signaling kinases. Why transcription factor NFB or nuclear factor kappa light chain enhancer of activated B cells family of proteins regulates various important cellular processes and influences genes involved in a plethora of biological activities which includes functions chapter summary. GPCR signaling, cell polarity, which mediates signal to hyposignaling through actin cytoskeleton. Why oxygen level is very important to maintain cellular homeostasis, and the hypoxia signaling pathway is involved in sensing oxygen level in almost all animal cells. While appropriate oxygen levels is important for cellular metabolism, but excess oxygen could be detrimental to the cells. Basically, the hypoxia signaling pathway is associated with almost all cellular processes, be it in epigenetic regulation or in secondary transcriptional cascades. HIF has shown to be involved in cellular growth, differentiation, immune, metabolic, or inflammatory signals. Why G protein coupled receptors (GPCRs) are the largest receptor protein family in humans. GPCRs share similar structural characteristics in 7 transmembrane domains and bound G proteins. GPCR activation by specific ligands is coordinated by several domains of the receptor in a complex manner depending on the ligand type. Why amplification of the initial stimulating signal through ligands to a large-scale biological response is achieved by activation of first and second messenger molecules within the cell specific to the pathway the receptor will be activating. Amplification of signal is crucial in cases of limiting extracellular stimulus, which is often countered in the body. Why G proteins are composed of alpha, beta, and Y subunits, and these have several structural characteristics. Covalent modifications of different subunits alter the functionalities of G proteins. Regulators of G protein. B from cellular proliferation to induction of apoptosis. Why JAK stator Janus kinase, signal transducers and activators of transcription is an important signaling pathway response to a range of growth factors and cytokines and hence translates the extracellular signal into transcriptional activation. This pathway is involved in transducing a multitude of signals in almost all multicellular organisms and helps maintain homeostasis, immunological development, and other developmental functions. Why WNTs? Wingless related integration site, signaling pathway composed of WNTs, which are secreted factors, plays a very crucial role in multicellular organism development. These secreted factors bind to frizzled receptors, which leads to regulation of various cellular activities like cell division, phase specification, patterning of the axis, other developmental processes like polarization, segmentation, and so on. Why Hedgehog signaling pathway is involved in cellular proliferation, differentiation, as well as in developmental processes wherein the pathway decides the cell fate and tissue patterning. Hedgehog signaling pathway is also involved in mature tissue homeostasis as well as maintenance of stem cells, tissue repair, and regenerative activities. The pathway is associated with various malignancies. Why Hippo pathway mediates the upstream signals to regulate biological processes downstream and coordinates with different biological processes like cell differentiation, death, growth, and proliferation. The upstream elements consist of G-protein coupled receptor nucleus because of the instability of actin oligomers. Why the tip nucleation model involves the foramen proteins complex present in the membrane, which mediates the filament extension stabilized by cross-linking by FASN proteins. The other model that has been suggested is known as convergent elongation model, which says that the ARC 2/3 complex is found to be essential for the initiation or nucleation of filopodia. Why the steady state phase witnesses the entry of the filament in a state of equilibrium where a balance is maintained in the disassembly of monomer from the end and polymerization at the plus end. This balance is maintained by critical cytosolic concentration of monomers of actin. This steady state assembly and disassembly of the actin filaments are known as treadmilling. Why a number of drugs from different sources have been found to play a major role in maintaining the dynamics of the actin filament. These drugs are very significant in the study of assembly and disassembly kinetics of actin. Why a highly concentrated three-dimensional network of actin filaments exists below the plasma membrane. The shape of the cell is actually determined and dictated by these very actin filament networks along with the associated actin-binding proteins. Why a number of protrusions and projections exist on the cell surfaces that are responsible for various cell movements like phagocytosis. Signaling or a group of different proteins involved in regulating G-protein-mediated cell signaling. Why adenylyl cyclase AC is an integral membrane protein that works as an enzyme to convert ATP to CAMP, which in turn works as a signaling molecule. AC can be activated by GPCR with its stimulatory alpha subunit upon activation. CAMP molecules bind to the regulatory subunits of protein kinase A (PKA) molecules and release the activated catalytic subunits of PKA. The catalytic subunits move into the nucleus and affect the transcription of several ligand-responsive genes. Signaling by CAMP is modulated by some proteins known as AKAPs, which bring together several regulatory proteins of the CAMP pathway in close proximity to one another in a cellular context. Why the movements of the cells are coordinated by the actin filaments in association with myosin. Here myosin is a protein that behaves like a molecular motor that converts the chemical energy to mechanical energy. Why when the actin filaments organize into a compact network, a structure called lamellipodia extends, forming a protrusion from the living cell. The protrusive movement of the cell is maintained by the quick assembly and the disassembly mechanism. Why the nucleation is the first step in actin polymerization. In this step, the actin nucleus is formed, which consists of about 3 of the actin monomers. This forms the basis for the elongation of the actin filament. The presence of G-actin monomers alone is not able to form the actin movement of the white blood cells towards the tissues in order to find an infection, metastasis of malignant tumors, etc., are a few examples of the movement of cells which involve crawling. Why cytoskeleton is essentially a complex system of interconnected filaments and tubules, extending from the center to the inner surface of the membrane throughout the cytoplasm. This means key functions in cell growth formation and carries membrane-limited organelles effectively in eukaryotes within the cytoplasm. Why MTs are the largest cytoskeletal elements in animal cells and are categorized into 2 subgroups, differing in organizational level and structural stability. The main group contains cytoplasmic microtubules. Why the microtubules consist of a straight longitudinal polymer called protofilaments. The fundamental subunit of a protofilament is protein tubulin heterodimer, 1 molecule alpha tubulin and beta tubulin each, and 13 protofilaments arranged side by side around the hollow center. Why doubles and triplets are composed of one full MT referred to as A tubule, 1 or 2 extra tubules known as B and C tubules comprising 10 to 11 protofilaments. Why AB tubulin subunits come together to assemble protofilaments. Protofilaments get associated with forming the roof of MT, and then the introduction of more subunits at the bottom of protofilaments extends the MT. Why microtubules have characteristic structural polarity. One end inherently develops or shrinks more quickly than the opposite end. B. Absorption of nutrients, etc. Most of these cell surface extensions are based on actin filaments, which are organized into either relatively permanent or rapidly rearranging bundles or networks. Why skeletal muscles are made up of bundles of muscle fibers formed by the union of many individual cells during development. Cytoplasm of the muscle cells consists of myofibrils, which are cylindrical bundles of thick filaments of myosin of diameter 15 nanometers and thin filaments of actin of diameter 7 nanometers. Why the sliding filament model for muscle contraction suggested that in the process of muscle contraction, there is a shortening of each sarcomere which results in the pulling of the Z discs closer to each other. Why in non-muscle cells, contractile assemblies of actin and myosin, which resemble small-scale versions of muscle fibers, are present. The actin filaments in these contractile assemblies are interdigited with bipolar filaments of myosin 2 just like in muscle fibers. Why myosin is the main constituent of the thick filaments, and each of the thick filaments is constituted by about 250 molecules of myosin. Why myosin is the main constituent of the thick filaments, and each of the thick filaments is constituted by about 250 molecules of myosin. Myosin has 2 significant functions, the first being the structural function where it acts as a building block for the thick filaments, and a practical one is where it acts as a catalyst in the breakdown of ATP during contraction and in its interaction with actin in the role of force generator of muscle. Why a variety of cells exhibit movements like crawling, which is the most basic form of cell locomotion on the surface of the substratum. The movements are shown by amoebas, YPTMs, like phosphorylation, palmitoylation, acetylation, the deletion of gene-encoded amino acids by detyrosination, and deglutamylation of alpha-tubulin or the addition of amino acids by polyglycylation. Polyglutamylation or tyrosination may result in few modifications. Why microtubule binding drugs are classified into 2 categories, microtubule stabilizers, such as apophylones and taxanes, and microtubule destabilizers, including colchicine and vinca alkaloids. Why these drugs have different locations on their target cell and are placed on microtubules depending on their binding locations. The taxane site prevails on beta tubulin, the vinca domain surrounding the binding site of beta tubulin GTP. The colchicine binding location is present at the junction between alpha and beta tubulin dimers. Why proteins that control microtubule dynamics lie into 2 major categories, proteins stabilizing the microtubules and microtubules destabilizing proteins. Microtubule-associated proteins MAPs bind to the microtubular wall at constant intervals, letting the interaction with filaments and other cellular structures and enhance MT stability. Why microtubule plus ends dynamics are controlled via several proteins affiliating with tubulins. Why the plus tip interacting proteins, also referred to as plus end tracking proteins, plus tips, are a unique class of proteins associated with microtubules. Why motor proteins are the major components of the class of molecular motors that can travel along the cytosol of animal cells. They change. Top of the microtubule, which is quickly growing, is regarded as the plus end and the other edge is the minus end. Why the assembly of microtubule is composed of 3 steps: the construction of protofilaments by AB tubulin subunits, combined protofilaments forming the MT wall, and elongation of microtubule by the conjunction of more subunits to the sides of the protofilaments. Why medicinal compounds can either inhibit the polymerization of MTs, destabilize MTs, and reduce the mass of the polymer MTs, or promote polymerization of MT, stabilize MTs, and increase the amount of polymer. Why development and shrinking of microtubules take place both in vivo and in vitro through the growth and reduction of biochemical AB tubulin subunits. Why a microtubule contracts rapidly through the change from reduction to development. This event is referred to as dynamic instability and can be utilized by cells in mitosis when chromosomes are separated. Why search and capture model is based on the dynamics of the microtubules in centrosomes and their nucleation. When microtubules develop in a discretionary manner from a centrosome, they look for the spaces while searching for kinetochores. Why post-translational modifications (PTMs) are powerful mechanisms for generating identities with microtubules and assist microtubules in various cellular functions like motility, cell shape, cell division and differentiation, intracellular signaling, formation, and transport of some specific organelles. Why microtubules also play a key role in mitosis. Three kinds of microtubules mediate mitosis: astral, polar, and kinetochore filaments. Why intermediate filaments are intermediate in size when compared to microtubules and microfilaments. Why they are composed of several types of monomers based on the cell type they are found in, except for the nuclear lamins, which line the nuclear membrane. Why the five major classes include keratins type 1 and 2, pimentin, GFAP, desmins type 3, neurofilaments type 4, and nuclear lamins type 5. Why the intermediate filaments go through several rounds of assembly till a compact structure is formed that usually remains polymerized but can easily exchange subunits from soluble pools. Why intermediate filaments are nonpolar and are not associated with motor proteins. Why intermediate filaments can be activated for disassembly by phosphorylation. Why organization of intermediate filaments are regulated by intermediate filament-associated proteins and by sequence determinants within itself. Why intermediate filaments have key functions like forming keratin networks, communicating between cells and matrix and muscle strength. Why intermediate filaments have key functions like forming keratin networks, communicating between cells and matrix and muscle strength. Why disruption of intermediate filaments can result in degenerative diseases of the skin, muscles, and neurons. Why microfilaments are formed by the polymerization of G-actins into two helices of F-actin. They exhibit polarity and are regulated by ATPase activity. Chemical energy into mechanical work with the help of ATP hydrolysis. Why there are three main kinds of cytoskeletal proteins: microtubules, intermediate filaments, and microfilaments. Why microtubules are made up of tubulin. Heterodimers, ab-tubulin dimer. Why they exhibit polarity and are regulated by GTPase activity. Why 13 protofilaments laterally associate to form a microtubule, with an alpha tubulin at the end and a beta tubulin at the plus end. Why a critical concentration of the monomer determines the polymerization state of the tubule. Why microtubules originate from a microtubule organizing structure called the MTOC, like the centrosomes or basal bodies. The end of the microtubule remains anchored in the MTOC. Why microtubules are seen in a dynamic state fluctuating between catastrophe, rescue, and steady state. A GTP beta tubulin cap is seen at the plus end of a microtubule, and the loss of which results in disassembly. Why several microtubule-associated proteins, MAPs, play a role in organizing the microtubules. MAPs like plus TIPS stabilize, while MAPs like kinesin 13 destabilize the microtubule. Why the key motor proteins of microtubules include kinesins and dyneins. Kinesins mediate a plus end-directed transport of cargo, while the dyneins mediate a end-mediated transport. Why cilia and flagella are made of microtubules with a characteristic 9+2 arrangement in the axoneme and a 9+0 arrangement in the basal body. Why the apoptosis induced by the external factor is called the extrinsic pathway. In the case, the stimulus for apoptosis is tumor necrosis factor TNF, an extracellular messenger protein. Why in certain cells of the immune system, TNF is produced in response to hostile conditions, such as exposure to ionizing radiation, viral infection, elevated temperature, or toxic chemical agents. Why in irreparable genetic damage, extremely high concentrations of cytosolic CA2+, viral infection, or severe oxidative stress trigger apoptosis by the intrinsic pathway. Why the activation of the intrinsic pathway is regulated by proteins of the BCL2 family of proteins. Why fate of any cell is determined by the BH3 proteins, whether a cell is going to the pathway of survival or death. Why BH3 proteins e.g., BID, BAD, PUMA, and BIM show their propoptotic effect in two different ways. Either they promote apoptosis by anti-apoptotic BCL2 members inhibition or promote apoptosis by propoptotic BCL2 members activation. Why it is vital for the health of a cell that BH3 proteins are either strongly inhibited or absent. The anti-apoptotic BCL2 proteins check the activation of propoptotic members. Why perforin molecule is secreted from NK cell, CTLs, which is transmembrane pore forming protein. The granules are serine proteases called granzyme. The most important component within the granules is granzyme A and granzyme B. Why mammalian cells gene called BCL2, an oncogene, contributes to the human B cell lymphomas. Unlike. Why actin polymerization is nucleated by formins, unbranched, and ARC 2/3 branched proteins. Why actins are organized by cross-linking. Proteins. Why myosin serve as motor proteins for microfilaments and can interact with their head domain on the microfilament. Their varying tail domain can give varying functions. Why in muscles, the actin filaments and the myosins are the key layers in the contractile unit. Sarcomeres. Why actin filaments and myosin 2 form. Contractile bundles. Why on activation, myosin heads walk along actin tracks, generating a force that mediates contraction in a CA2+ dependent way. Why myosins I, V, and B mediate intracellular translocations by walking processively on actin filaments. Why program cell death is vital regulation of cell number as well as cell health in an organism. It is an energy-independent process of cell death. Why necrosis or apoptosis pathway is dependent on the signal, the tissue type as well as the developmental stage of the tissue. Why necrosis is uncontrolled, which affects a large number of cells. On the contrary, apoptosis is a controlled process which affects only an individual or clusters of cells. Why various biochemical modifications are associated with apoptosis like protein cleavage, protein cross-linking, breakdown of DNA and phagocytic. Why caspases are widely expressed in most cells as an inactive proenzyme form. Once activated leads to the initiation of a protease cascade. Why innate immune responses constitute the first line of defense against external pathogens and employ germline-encoded recognition molecules in phagocytic cells. The response time for innate immunity is fast but is much less specific compared to adaptive immune response. Adaptive immune responses take days to develop but are much more specific and potent than an innate immune response to pathogens. Why activation of the innate immune system relays signals to stimulate and direct the subsequent adaptive immune response. The two arms of the vertebrate immune system, therefore, act cooperatively. Why adaptive immune response depends upon antigen-specific cell surface receptors called T cell and B cell receptors, which are randomly generated in B cells and T cells during maturation. Why once engaged by antigens, individual T and B cells undergo clonal selection to produce a homogeneous population of antigen-specific, reactive cells. Why residual B and T cells from an antigen exposure or immune response form memory T and B cells, which help mount a much faster, highly specific and highly potent adaptive immune response in case of secondary infection by the same pathogen. Why all red and white blood cells are developed from pluripotent hematopoietic stem cells through a highly regulated process known as hematopoiesis. Why hematopoiesis takes place in the bone marrow in adult vertebrates. Other oncogenes proteins, BCL2 is found to inhibit apoptosis. Why apoptosis is a tightly regulated signal cascade including various pathways. These different molecules involved in the cascade can be used as a marker for apoptotic assay, e.g. DNA fragmentation, detection of caspases, membrane alterations, mitochondrial assays. Why the state of protection against foreign pathogens or antigens is known as immunity. Vaccination is a procedure through which the immune system can be prepared to mount an immune response quickly, thereby eradicating the pathogen and the disease before it can cause widespread epidemics. Why humoral immunity is mediated by antibodies, which can be found in bodily fluids and are produced by B cells. Passive immunity can be provided to individuals against specific pathogens by the transfer of antibodies against them from one person to another. Why cell-mediated immunity primarily involves cytotoxic T lymphocytes, which help in the stimulation of other immune cells, eradication of infected cells and removal of pathogens. Why once infected, the immune system adapts to the foreign pathogen quickly. Such a response relies upon antigen-specific recognition molecules that can be germline-encoded or randomly generated. Why the phenomenon of self-tolerance limits the immune system from attacking host tissues. Why the vertebrate immune system has two interconnected facets, innate immunity and adaptive immunity. Expressing T cell receptors can have NK cell-like features and are termed NKT cells. Why regular or non-specific immunity is the shield structure with which an individual is considered. Most sections of this safety are accessible before the start of an ailment. Phagocytic cells, antimicrobial blends accept a noteworthy activity in characteristic opposition. Why the complement is a course of approximately 30 serum and cell surface proteins or glycoproteins, which expect critical employment in both natural and versatile resistant reactions. Essential components of enhancement structure are lysis of cells, opsonization, viral equalization, combustible response, solubilization and opportunity of a safe complex from the course and stored them in the spleen and liver. Why the inception of the complement by the classical pathway begins with the game plan of antigen, antibody, A-flat complex or the legitimate of the immunizer of the suitable huge antigen, for instance, a cell inside seeing enhancement protein. Simply surface-bound IgM antibodies can sanction supplement, and they do in that capacity by methods along these lines. Why the alternative or properin pathway of complement activation moreover gives self-sufficient authorization of enhancement on pathogen surfaces. This pathway is in a reliable state of low-level inception, known as takeover. Why the lectin changes from the classical pathway just in the hidden affirmation and incitation steps. Without a doubt, it will, in general, be thought that the lectin pathway. An environment that supports both the self-renewal of stem cells and their differentiation into multiple blood cell lineages. Why two main blood cell progenitors are formed due to the differentiation of hematopoietic stem cells, common myeloid progenitor cells and common lymphoid progenitor cells. Why common myeloid progenitor cells give rise to four main types of cells: erythrocytes or red blood cells, monocytes which in turn differentiate into macrophages and myeloid dendritic cells, granulocytes such as neutrophils, and the less abundant basophils, mast cells and eosinophils, and megakaryocytes. Why macrophages and neutrophils are major effector cells of the innate immune system and specialize in phagocytosis and antigen degradation. Additionally, macrophages can also present antigens to T cells in association with T cells. Why dendritic cells are the most potent antigen-presenting cells of the vertebrate immune system and most effective in stimulating naive T cells to activation. When immature, dendritic cells can capture antigens at one location, undergo maturation, and migrate to another location to present antigens to T cells. Why B cells, T cells, and natural killer NK cells are the three types of lymphoid cells. B and T cells harbor antigen-specific receptors with unique specificities and are members of clonal populations. T cells display antigen-specific T cell receptors on their surface, while B cells express antigen-specific membrane antibodies on their surface. NK cells do not express any antigen-specific antibodies. A small subset of T cells. Why macrophages and monocytes are a bit of mononuclear phagocytic structure. Macrophages are capable for the root doing phagocytosis. Why neutrophils are furthermore called polymorphonuclear neutrophils (PMNs), which structure the colossal masses of leukocytes because of their sporadic shaped centers. Why cells present in tissues that catch antigen and migrate to the lymph center points and spleen, where they are particularly unique in demonstrating the ready antigen to T cells. Dendritic cells can be gotten from either lymphoid or mononuclear phagocyte genealogies. Dendritic cells gain antigen by phagocytosis. The antigen is arranged and creates dendritic cells present it to TH cells. Why basophils are non-phagocytic granulocytes. They expect a critical activity in red-hot and excessively touchy responses. They discharge histamine, serotonin and heparin, leukotrienes, prostaglandins and have a receptor for IgE. Why eosinophils are motophagocytic cells and are antiparasitic and antiprotozoal in nature by releasing cationic peptides and responsive oxygen intermediates into the extracellular fluid. They help in repairing wounds and some extreme touchiness reactions. Why natural killer NK cells express cytotoxicity against a wide grouping of tumor cells and disease-tainted cells. They build up lymphocytes in the human periphery blood. NK cells don't possess film receptors to perceive B and T cells. NK cells perceive their target by recognizing. Should not be seen as an alternate pathway but rather a course for classical pathway incitation that avoids the prerequisite for AB. Why the terminal fragments of the complement falls are C5b, C6, C7, C8 and C9. These parts bind to each other progressively to outline a macromolecular structure called film salt complex or MAC that results in cell lysis. Three enhancement pathways consolidate MAC. This puzzling structure is a huge channel through the film of the goal cell, engaging particles and little atoms to diffuse uninhibitedly over the layer. Why inborn immunity, additionally called common or local immunity.